This method can determine both purified and non-purified AAV vec titers, and assess the ability for the AAV-AAP to promote assembly of cognate and heterologous AAV serotype capsids. The main advantage of this technique is that it is a straightforward, inexpensive method that avoids the drawbacks of other AAV titration methods.
Though this method has commonly been used for AAV vector quantitation, it can also be applied to address various fundamental questions about AAV biology as shown in this protocol.
On day 0 after culturing HEC293 cells to 90% confluency, prepare the transfection reagent by mixing the plasmids in 96 microliters of PBS without calcium or magnesium in sterile 1.5 milliliter microcentrifuge tubes. As indicated in this table, the total amount of DNA is 2 micrograms per well. Add 4 microliters of PEI solution to the PBS plasmid DNA mix. The final volume is approximately 100 microliters, or 5% volume of the culture medium. Vortex the tubes briefly and incubate the DNA PEI mixture at room temperature for 15 minutes. Following the incubation, briefly spin the tubes in a microcentrifuge to collect the liquid to the bottom, and add the DNA PEI mixture dropwise to the culture medium in each well of the HEC293 culture plate. Then gently agitate the plates and return them to a 37 degrees Celsius incubator with 5% CO2.
Transfection